Alright, so a couple of you have asked me to put some notes online going through this activity. So I'll do my best, and please just ask questions. So I want to go through diffusion, osmosis, and then active transport. So I'm going to start with diffusion. So here we have the cell membrane, obviously, and we're going to put a high concentration of carbon dioxide outside the cell and a low concentration inside the cell. I'm just going to run it for a second and then stop it so that we can get our starting levels. So we're not worrying at all about the O2. We can just cross that out. And we're only looking at the CO2. So you can see outside the cell we have a high concentration of carbon dioxide, and inside we have the low. And if you look, right, we talked today about concentration gradients. You can see like this would be the concentration gradient. And the steeper that concentration gradient, the faster you're going to move towards equilibrium. You'll notice as it gets closer to equilibrium, that rate is going to slow down because you're going to have particles going in both directions through the membrane. The particles are moving randomly, and they'll only stop moving in one direction when they hit something else. Which means you're going to have some particles moving this way through the membrane and some particles moving this way through the membrane. But just the fact that there are more particles outside the cell means there are more opportunities for them to bump into things, which means there's a greater chance they're going to move through the membrane inside the cell, which is why you end up with the net flow being in towards the cell, because we're going from high concentration to low concentration. So let's go ahead and run that, and we can see those are evening out, right? That gradient is getting shallower and shallower. Now, one thing I would encourage you to do is just pick one of the particles and follow it. Watch how it goes in a straight line until it hits something else, or until something hits it. And you can see that these really are just moving randomly. You can also, if you look at the membrane, if you focus on that for a little while, you can see, oh yeah, here's one moving that way, there's one moving this way. They are truly going in different directions. So that's diffusion. It's just this random movement, but it's random yet predictable in that, based on the fact that things are going to bump away from each other, and where there's a high concentration, there's going to be more bumping. You're going to get a predictable flow of high concentration to low concentration. So that's diffusion. Some of you have asked about facilitated diffusion. That's a little different, and if anybody's interested, I can talk to you about that one-on-one in class. But suffice to say, there are more ways to get through a membrane, facilitated diffusion being one of them, than just this random diffusion, simple diffusion, osmosis, and active transport. So just be aware, like anything else, we're getting part of the picture. We're not getting every single detail because there's just too much out there to get every single detail, and we want to get big picture. Okay, so now we're going to look at osmosis. And I'm actually, let me move this down a little because I don't like their definition. I find their definition actually pretty incorrect, where they're saying osmosis is simply diffusion of water. It's not. Osmosis has to do with this idea called osmotic pressure. So what this here is saying, this is a membrane, but now you'll notice all these little kind of burbly things on the membrane. Those are aquapores. And so what this is saying is there's so many aquapores in a membrane that water basically can move freely across the membrane. It does have to move through an aquapore, but there's so many aquapores that it can move pretty freely. So let's put a high salt concentration outside the cell and a low salt concentration inside the cell. So the water being blue, because we're always going to color the water blue, and then the sodium and chlorine, or chloride because it's an ion, from the salt being red and yellow respectively. And there should be, I mean, you're welcome to pause the video and count, but why waste your time? There should be a 1-to-1 match of yellow to red because sodium chloride has 1 sodium for every chlorine. And ionic compounds, sodium chloride being one of them, has this unique feature where when it's dissolved in water, it's actually, its ions are divided up. They're no longer touching each other like when it's in solid form. This isn't liquid. It's not that the sodium chloride is liquid. It's what we call aqueous, and it's often abbreviated as AQ, and that means dissolved in water. I'm more than happy, again, to talk to anybody who wants to about what it means to be aqueous. This is one reason why salt water conducts electric charge, because going back to that active transport idea that we need to separate the charges to get a current to flow, once the positive and negative ions are what's called dissociated, but separated because they're dissolved in water, then an electric charge can flow, which is why salt water conducts electricity. Pure water doesn't actually conduct electricity, but most water out there has some salts dissolved in it. All right, so again, I'm going to run this just quickly and then we're going to stop it so that we can see what's going on. So here we have two ideas. We have osmotic pressure, which is here, and then we have the salt concentration, which is here. So obviously the outside of the cell has the higher salt concentration. That leads to a higher osmotic pressure because osmotic pressure is based on how much stuff is dissolved in the water. The more stuff dissolved in the water, the higher the osmotic pressure. So on the inside of the cell membrane, where you have a low salt concentration, you have a low osmotic pressure. And notice here we're not talking about the water. We're not talking about concentrations of water. We're talking about concentrations of stuff dissolved in the water. And yet it's in osmosis, it's the water that moves. You're not going to see any of the sodium or chlorine going through the cell membrane. You're just going to see the water going through because these are aquapores. And aquapores, if you want to get into the details, actually repel charged particles. And sodium and chloride are both ions, so they both have a charge, sodium being positive, chlorine being negative. So it's the water that's moving, even though it's the salt that's causing the pressure. So that's where people get confused because they want to link this to diffusion. And they're like, well, in diffusion, stuff moves from high concentration to low concentration. But in diffusion, it's the stuff we're talking about the whole time, right? We're talking about a high concentration of carbon dioxide, for example. And so carbon dioxide moves away from that high concentration of carbon dioxide. Here we're looking at the movement of the water based on the concentration of salt. So the water is moving from a low concentration of salt to a high concentration of salt because it's trying to make this high concentration more dilute. And so let's run that for a moment and see what happens. Okay, now first of all, predict what's going to happen to these pressures. Okay, right now we've got low pressure inside, high pressure out. We're going to expect the water to move from dilute to concentrated. So let's run that. Okay, so we can definitely see water moving through. Notice there's no red or yellow moving through. But one thing to notice is that the pressures aren't actually changing that much. So let's stop this. And one of the reasons the pressures aren't changing is because there's an actual movement of water, which are particles, which changes the overall pressure inside. As water moves out of the cell, you have less water in the cell, so that means there's lower pressure in the cell. That's why when we're looking at the movement of water, and we saw this with our cheek cells, the membrane actually moves. Okay, now what happens is the cell swells, the cell shrinks. Here it looks like almost a wall moving, but remember that if this were a membrane around an entire cell, that's the whole cell expanding or the whole cell contracting. So we're going to say, we're going to put in the movable membrane here, and now we're going to run it. And as that water is leaving the cell, let's stop it for a second because it's telling us what's going on here. the cell is shrinking. This is exactly what happened when you put corn syrup outside your cell. Instead of sodium and chlorine, you had sucrose dissolved in the water, and so water left the cell and the cell shrunk down, just like this, because water is leaving faster than it is entering. So we can run that for a little, and now we're starting to see those pressures equalizing as the water leaves the cell and the cell gets smaller. Because now, even though there's more stuff outside the cell, it's a larger space. And then we can run that in reverse if we wanted. But let's, in this case, put a low concentration here, and we're going to put a high concentration here. We're going to keep the movable membrane, let that run. First of all, those immediately switch, right? But now we see the cell's expanding because water is entering the cell faster than leaving. If we take out the movable membrane, well, it's going to stay where it is right now. Had I reset it, let's reset that, and now run it without the movable membrane. Notice all this water is entering the cell. That's causing a much higher pressure inside the cell simply because there are more particles. And pressure is a result of how many particles are inside the cell at any one point in time. All right. So now we get to the active transport part, right? And this is where we want to create an electric potential. And I'm just going to go through this really quickly because it seemed like most people were good simply with the creating of the electric potential. So the idea being, if I want an electric potential, I've got to separate my charge. I've got to get my charge different on either side of the membrane. So moving some positives from one side to the other, moving some negatives from one side to the other. By the way, most of the place you get your ions, so these are ions, is in the foods you eat, in the salts in the foods you eat. And most of the time that's, oh, I shouldn't say that. A lot of the time that's sodium chloride. But actually in chemistry, any compound that's ionic is known as a salt. So one of the things I would say is you can hide the water on this. A couple of people said, like, why is the water there? The water is there simply to show you that these ions are dissolved in water, right? We've talked about how water is very unreactive, but people have asked them, you know, what do we use it for in our bodies? All of these things are dissolved in water. All of these reactions take place in an aqueous solution. I just talked about that a few minutes ago. That aqueous means dissolved in water. So you can hide the water to make your life a little easier if you want. I'm actually going to leave the water here because I think it's fine and you'll live. But now we see we have an electric potential. And nothing is moving anywhere. And then again, if we stop that, up here, actually, let me see if I can highlight. Ooh, pretty. Here, this is the part that's talking about why you need that electric potential. Electricity is what allows you to move, whether that's moving your muscles, whether that's sending signals up through your nerves, whether it's, you know, allowing your cells to control the flow of water through osmosis. And so that has to do with the expanding and contracting of the cells, which goes back to the osmosis we just talked about. It's what allows, your heart is a muscle. It's what allows your heart to pump. Everything you do has to do with well, not everything, but much of what you do has to do with running an electric current across a cell membrane. And you can only run an electric current if you have voltage, if you have an electric potential. And the way you get the electric potential is by separating the charge. But that doesn't happen naturally. You need energy to make that happen. Osmosis happens without an input of energy. Diffusion happens without an input of energy. But you'll notice we're going, as we said before, against the concentration gradient. We're trying to put more negatives where there's already negatives. We're trying to put more positives where there's already positives. So we're moving against that gradient. Or if you're looking at positives, right, we're moving positives inside the cell. If this is our gradient and we're trying to get more inside, then we've got to be going up this gradient. That's the opposite direction. All right, and so finally we get to, we understand why we need the voltage, the electric potential. Now how do we get it? So one of the things we use our ATP for is to change the chemical energy of the ATP into, as we said earlier, kinetic energy. Kinetic energy is energy of movement. Now this is, I just want to point this out, load model, right? This is a model. This is a picture that's going to help us understand what's going on. On the last page we could hide the water, or we didn't have to. Here the water's hidden, but there's also water. There's also carbon dioxide and oxygen, right? Think back to that drawing you made of the cell membrane with all those different protein channels. All that is going on all at the same time. This is simply looking at, okay, let's take one ion, calcium, and see how we can move it against the concentration gradient. And let's just focus on that for right now. So I'm pretty much going to do exactly what I did in class to show you this. And as I said, this is one thing ATP is used for. ATP, some people asked in 4th period, what else do we use ATP for? And I completely drew a blank, which is funny because I just talked about how ATP is used in the light-independent reactions of photosynthesis. It's also used in cellular respiration to provide more energy, right? We talked a little bit about that, or maybe we didn't. I can't remember. But you use some of the ATP you produce in cellular respiration simply to run cellular respiration. Any endothermic reaction in your body requires ATP. And so any energy your body needs to run any reaction that requires energy, that energy is coming from ATP. So that's everything, that's all the stuff we use ATP for. To copy and synthesize DNA requires energy. That's using ATP. If your body needs energy for something, it's ATP. So let's take a look at this. And again, I'm going to stop it at times. So here, just quickly, we have an equal amount of, or equal concentration of calcium inside as outside the cell. We've got a lot of ATP, so a lot of chemical potential energy here. This is the protein channel. We've got a binding site. The reason is because the calcium is still moving randomly, just like the other particles we saw earlier. But once it gets into the channel, we want it to stay in the channel because we then want to move it someplace else. So we want to hold on to it. So it literally binds to the protein channel. And later this year, we're going to talk about the structure of proteins and how they work. But think back, we already talked a little bit about enzymes. Enzymes are also proteins. And we know enzymes have these active sites that certain molecules fit in so that they can either be synthesized, brought together, or broken apart. Right? So this is simply like it's a binding site out of another protein. This isn't an enzyme. This is a protein channel. But they're both proteins, and the way proteins are made allows them to bind to certain other molecules. Those of you who've looked at facilitated diffusion, by the way, those are usually carrier proteins. Those are carrier proteins that are used there. All right, so let's go to run. And, oh no, our calcium just went out. Now there's a big ATP blocking the way. Here comes another calcium. Nope, that one went out too. Here comes another. I have high hopes. Nope. Oh, wait, it's going back in. We're waiting. Okay, there we go. So now the calcium is connected to the binding site. So now right here on the protein channel, the protein is actually going to change its structure so that ATP can bind to it. And I'm really bad at stopping it at the right time, so I'm just going to do my best. So there you can see that forming. Now there's a bunch of calciums in the way. Here comes an ATP. Okay, I'm going to stop it there. I'm going to take a guess that it's this ATP that's going to react. Fingers crossed. So remember, when ATP goes through a reaction, it becomes ADP plus P and energy is released. It's a hydrolysis reaction with water. Don't worry about that reaction. Again, that's something I can give you resources on if you're interested. Some of you are still very bothered by this ATP to ADP thing. Others of you are very happy knowing when ATP goes to ADP plus P, energy is released. And chances are I am not going to be able to pause it right when the energy is released, but look for like a little starburst of, ooh, energy. Nope, it wasn't even that. Oh, there we go. Yep, see, I just missed it. But there was that little starburst of energy, and now we have, here's the ADP plus P that was the ATP. And now notice the protein channel has started to open. So what that energy did, it was chemical potential energy. It's now, that chemical potential energy is used to move the protein channel. So that's why we talked about chemical potential becoming kinetic, because the protein channel is moving. And so that moves, and you'll also notice now the chemical potential energy from before, this is where we started, has dropped a little, and the electrical potential has gained a little. And so now that calcium kind of bumbles out there, and now we wait for another one. The protein channel goes back. There's another one. There's our binding site. Okay, release of energy, calcium goes out in the other direction, and we're now increasing that electric potential, and so on and so forth. And so that is how we get that electric potential. We know we need it so that we can conduct current, so that we can do many, many functions in our body. All right, hopefully this is more helpful. I would point out that I don't think you need me to talk you through this. I really think that if you sat and looked at it and worked with it and read what it said and maybe asked some questions, you could come to this on your own. That's a skill too, is saying, okay, this is kind of confusing. Let me look at this again. Let me stop it again. Let me really, let me stop it when there's a message, right? We've got this messages on thing. What is this? Okay, it's waiting for that. And oh, now I see that site is forming. All I've done is read what's on the page for you. So please ask questions in class, but please try to understand what your questions are rather than waiting for me to point out that you're confused. Have a good night.